Good morning. Today, inshallah, we'll start with lesson 3 in chapter 12, which is the DNA replication. In this lesson, we will understand how the DNA replicates itself, the process, the enzyme, how the base pairing will form in this DNA replication. So my question today is, how does DNA copy itself? From this question, I can conclude that I need to understand the DNA replication as an objective. So in the beginning, what we mean by DNA replication, what and why? So DNA making a copies of itself. So again, what we mean by replication, DNA making a copies of itself. Why they need to copy itself? DNA must copy before a cell can divide, because we need the DNA for the cell and when it is going to divide. Each new cell will have a complete set of DNA. So for the parent cell, it's having here 4 chromosomes. Before the parent cell will be divided into 2 daughter cells, I need to duplicate the DNA. And why I need to duplicate the DNA? To obtain 2 daughter cells with full set of chromosomes. Let's check what you mean by replication and how the scientists discovered the replication process. In the beginning, Watson and Crick realized that complementary base pairing provided a way for the DNA to copy itself. So we have a complementary base pairing here, and we have the sequence for the DNA already here. Base pairing could allow a new strand to be built on an old strand. So we can form a new strand here if we have one old strand. So we will make a base pairing. So there is many models for the DNA replication, and the scientists make many experiments to check what is the possible or what's the correct replication model. The scientists conclude that the best replication model is the semi-conservative model. What do you mean by semi-conservative replication? Semi-conservative replication, halfly conservative. It means that the newly formed DNA will have one old strand, or what we call it original strand, and one new strand. So again, they mean by semi-conservative replication that the newly formed DNA must have one old strand, one new, one old, one new. Each DNA molecule must have one old strand and one new strand. So each strand in DNA molecule is used as a template to build a new strand. This is the template for this one, and this is the template for the other one. Okay? For the DNA replication process, you have to know that the replication needs many enzymes to finish the replication. So replication begins with an enzyme called DNA helicase. It's not important for you at this stage, but you have to know that there is enzymes will open the DNA, forming something called replication bubble. This is the replication bubble, okay? Once you reveal or open the DNA, you are exposing now the 2 strands for another enzymes to come. And once the other enzymes comes, the DNA replication will start. As you can see here, the replication bubble will open the 2 strands. That means we have 2 directions for adding the DNA. You have to know that. You have direction for unwinding here, and at the same time, the the 2 DNA strands will be open to 2 replication formation. Okay? There is a link here. You can use it to watch the replication process, and there is a simulation also. You can use it to check the replication process. Multiple replication bubbles are open simultaneously, allowing the molecule to be copied or replicated quickly. For the original DNA, there is multiple replication point or multiple replication fork that will be opening to form the replication bubble. And as you can see here, the DNA will replicate itself until it's fully replicated. In the process, the nitrogen bases on the original DNA strand are exposed in the replication bubble. They will serve as a template to build a new DNA strand. So in the original one, this is an original strand, and this is an original strand. And here, the nucleotides are exposed, and here the nucleotides are exposed. And once the enzymes is here, the DNA or the enzymes will start to add a complementary sequence to complete the newly forming strands. Again, this is the replication bubble, and here we have the replication fork. For each replication bubble, we have 2 replication fork will begin. After the helicase, DNA polymerase begin or bring a nucleotide to the replication fork. So there is a replication fork which needs an enzymes. The enzymes are, there is many enzymes will work. Before the DNA polymerase, there is a primase will add the primer, and the DNA polymerase will find the primer and bind to it, and then it will add a new new nucleotide to the replication fork by the form of base pairing. So each A will take T, and each C will take G, and vice versa. Each T will take A, and each G will take C. You have to know that the replication must be into the 2 strands. So one of the strands, which is 5 to 3, you have to know that's the leading strand. And the other strand here, from 5 to 3, it's known to be lagging strand. So we have 2 strands, one leading strand and one lagging strand. From the direction, this is 5 to 3 in this side, and this is the direction from 5 to 3 to the other side. So this side, like this is the leading strand, the other is lagging strand. What do you mean by leading strand or by lagging strand? The leading strand replication will be continuous replication. So it's built toward the replication fork. So the direction will be to the replication fork direction. So look at this direction. This is the replication fork. The direction here will be perfectly to the replication. So the replication will be continuous. While here, this is the replication fork, and the direction is to the other side. So the replication will be discontinuously. So in the lagging strand, the replication occurs in short segments called Okazaki fragments, and the new DNA strands grow away from the replication fork. So there will be small discontinuous Okazaki fragments, and once the replication finishes, the Okazaki fragments will seal together to form a full DNA. Again, each bubble has 2 replication fork. You have to make sure that the lagging strand must replicate it in the same direction of the leading strand. So the lagging strand will be discontinuous strands or forming Okazaki fragments. Okay? The process of the DNA replication continues until 2 complete copies of the DNA are produced. So at each replication, 2 DNA molecules, 2 complete copies of DNA molecules will be formed. Each copy of the DNA has 1 strand of the DNA from original and 1 a newly formed strand, which is again known as semi-conservative replication. Again, this is the DNA. The replication fork started, the replication bubbles formed, and here another enzyme will make a replication. This is the helicase, which open the DNA, and it's completing the opening, and the DNA polymerase will add here the newly nucleotides on the old strand to form newly DNA, and here we have 2 completely formed DNA molecules. Each molecule contains 2 strands. One of these strands is old strand, the newly one is the new strand. So each molecule contains 2 strands, one new and one old. This is what we call it the semi-conservative model. This is a link for a simulation. Okay, this is a simulation for DNA replication process. You have to watch this video and you have to watch the simulation process to understand perfectly the DNA replication. You have to know that at the tip of the chromosomes, at the tip of the DNA, there is a region called telomeres. The telomeres, which is the tip of the chromosomes, are known to be telomeres. If you look at the picture here, the yellow part of the chromosomes are the telomeres. Over the time, DNA may actually be lost from telomeres. So each time a chromosome must replicate, it will lose a small part of the telomeres. So there is another enzyme called DNA or telomerase. The telomerase solves this problem, which is the shortage problem. This enzyme will add short repeated DNA sequence on the telomerase. Okay, so the telomeres will be elongated by the action of the enzyme that's called telomerase. At the end of the lecture, you have to know that the replication differ between the prokaryotic and eukaryotic. As we know before, the prokaryotic, which is the cells that lack a nucleus, while eukaryotic cells, the cells that contain the nucleus, and the DNA is found inside the nucleus of the eukaryotic cell. And you have to know that in the eukaryotics, the DNA is long DNA, and at the same time the DNA is double helix DNA, while in the prokaryotic cells it's a circular DNA, okay? So prokaryotic the DNA replication starts from single point and goes into two direction until the entire chromosome is copied, while the eukaryote replication begins at dozens or even hundreds of spots on the DNA, going into both directions until each chromosome is completely copied. So here in the eukaryotic, there is multiple replication forks, multiple replication bubble, and here in the prokaryotic cells or the prokaryotic DNA, there is just one replication bubble with two replication fork. You have to understand that the prokaryotic DNA is a circular DNA, while the eukaryotic DNA is longitudinal DNA. Okay? Till now, the replication process is finished. Have a nice day. Thank you.